you all see this slide? Super. Thank you very much. So, I will be giving the presentation, and I will take questions. So if you have any, please ask them in the chat or in the Q&A, and then I will review them after the talk. And if I can manage to find, I'll unmute. Maybe we can you can ask the questions in person. So thanks in advance. So today, so again, I would like to present Vesiculab activity, and the title of the talk is Advancing the Extracellular Vesicle Workflow. As you may know, I've been personally involved in the field of EVs for about 15 years now, working for first Eisen Science, then Nano FCM, so being very involved in the analytical side, and as well as the sample preparation for extracellular vesicles. And back in June 2023, I started in the Vesiculab, so which is my own company based in Bestwood Village in Nottingham, UK, with humble beginnings, but now we are fully fledged team with lab production and operations spanning sales spanning 5 continents. We mainly focus on reagents and consumable solutions for research on extracellular vesicles, also as other nanoscale entities such as nanomedicines, LNPs, and viruses. But today I will focus on extracellular vesicles to make the talk not too long. What's very important to me is to bring solutions that are innovative and really accelerate the research, as opposed to have one iteration of each and every possible techniques that you may use in the lab. So fast tracking is really important, because over the years I realized that this was really linked to improvement of research. The studies need to be bigger, and they need to be faster to get the full potential of extracellular vesicles. And the picture here, that's where we are, in that ancient building in Nottinghamshire, and the team and the project students that come to join us based on the projects. Oh, sorry, I think that we have more people trying to join. Sorry about that. So this slide is really to illustrate the workflow that researchers face when they want to study extracellular vesicles, and it's illustrated on the left, and then there's a chart on the right. But mostly the starting material will be a fluid that contains either cells or bacteria, or an entity that excretes extracellular vesicles. And then the first step will be to isolate these EVs and enrich them, as opposed to leave them with their mother entity. So isolating from cells or bacteria. Originally, that has been done by ultracentrifugation mostly, also density gradient, and more recently with size extrusion chromatography, as we will discuss. And the characterization is very important to assess the purity, to assess if they are isolated. 15 years ago, that was really the case, that we didn't know if it was if there were any EVs at all, to see their physical characteristics as well as their phenotype, what biomarker do they contain. And finally, the goal is to discover the function and the function or the properties of the biomarkers that they're carrying. So we want to see how they are uptaken by cells, or maybe discover if the biomarkers that they bear are going to be indicator of a condition of a disease, etc. So that chart is really is summarizing these illustrations. I added stabilization because I think it's very important that post isolation to secure the EV so that they don't deplete with time. And the traceability is really to make them stand out. It's not always possible to do label-free analysis in order to get their characteristics and the functions. And again, the goal of Vesiculab is to reduce the time that it takes to do the whole workflow, as well as minimizing materials lost. So let me, sorry, I put another, someone else want to join. So the first part of the presentation will be dedicated to our solutions for isolation and purification. And the focus is on size exclusion chromatography. Is potentially because I have been personally involved with these techniques, but over the years I came to really believe that this was the most suitable for extracellular vesicles isolation. Its principle is that it's the sample is added onto a bed of resin that is made of these beads that are porous, and the porosity is such that when nanoparticles are applied to them, they are going to either go within these pores and basically being delayed according to the size of the object, or not going at all means that they are excluded, like the beads in the particles in green. So when exclusion is happening, the product is eluting very rapidly and is eluted first. And when the particles are in the range of the porosity of the pore size, then they are going to be delayed, and the separation occurs between the particles that are excluded and the one that are resolved. The pros of this method is a gentle isolation. The phenomenon doesn't involve any particular pressure or chemical process, so there's no harsh treatment of the sample being isolated this way. It's compatible with all fluids that contain extracellular vesicles, and the yield is quite decent. So we need to expand a little bit on what it means. It's a technique that has been acknowledged as let's say accepted by the community, including in the MySafe guidelines that have been drawn by the ISEB. So that's the pros. Let's say the cons of this technique when it's done by gravity is that it's fairly slow. You need to wash the column, you need to collect fractions, and these fractions are basically separating the different population, and by not combining enough fractions, then you end up losing some of the material. So it's always a matter of yield vs purity. You have to compromise. Do you want maximum particles or do you want maximum purity? And the EVs themselves are not resolved by size, so they are excluded from the gel, but you don't really have the possibility to isolate different population of EVs by size, not with with a normal gravity column, you need to have a very, very long side-screen chromatography column, and probably with a low pressure system type of actor, for example. So that's despite all these columns have been quite popular over the years. I'm sure you have been using them at the moment. So what we wanted to do at Vesiculab is to take the view that if it wasn't really possible to use, if side-screen chromatography was used to mainly remove impurities from extracellular vesicles, so removing all the cells, sorry, the protein that are contained into the samples are small size. So we took the view that in order to do that, it wasn't necessary to do a long collection of fractions. So we went for a model of spin column. Rather than adding the sample and collecting fractions, the gel is prepared by centrifugation 1 minute at 1000 G, and the sample is added and spun himself for one minute at 1000 G. And what this does is to yield the EVs purified from small size impurities. So the operation is in gentle, 1000 G. That's less than what you are using to remove cells from EVs. It's just 1 minute per for the isolation itself, and two steps for the preparation of the column. And the interest is that it's really scalable. Here I'm showing the midi size column that are included into normal size centrifuge. There are much bigger centrifuges. Even for 15 milliliter tube, you can find centrifuges that can hold more than 100 samples at a time. And these centrifuges are really common. You have tens of them in the lab. And the interest is that there's no fraction collection, no dilution of the sample. There are two models. One is for 50 to 100 microliters load volume. That's the micro model, and the mini model is for 0.5 to 1 ml. The exclusion limit of the gel, or the Corcept gel, is a 35 nano. So it means that whatever is larger than 35 nano is excluded, and whatever is smaller is retained on the gel. That's the theory. In reality, there's always a zone, so it's like a band. So that's why we tend to recover EVs that are really from 50 nanometers. Typically, that's limited by the capacity of measuring them anyway. And smaller size entity travel through the gel, but they don't elute because they're captured on the gel. And for these reasons, it works very well with both cell cultures and biofluid. We recently run some samples for clinical studies, urine samples, and they perform very well. It's designed for single use, and it's a matter of regenerating the column, because it's done on a spin action. You can't ensure that you are going to release the impurities from a very dirty sample. So that's it is single use, which means that you don't have any carryover from one sample to the next. And as you see in these graphs, the recovery is very, very good. It's at 90% for the nanoparticles, which is very efficient, 90% plus. And partly because it's non-dilutive, it's very efficient in itself, and it's non-dilutive compared to gravity sec columns. And typically, we're talking about 111 particle per ml concentration as a maximum load. That depends very much on the purity of the sample. But to give you an idea, and I explain later, we can the vis columns can handle plasma. Depends on what experiments we want to do. And talking about plasma, we are launching a new kit which is based on the Visisec columns. It's called Vesiomics, and it's as the name entails, it's designed to isolate EVs from plasma to perform mass spec experiments. So as you know, mass spec experiments are very sensitive to not only proteins but lipoproteins, and these co-elute with EVs during side-screen chromatography. So it's important to remove them for good mass spec, but there's a lot of them if they're not specifically removed. In normal conditions, particles in plasma will be more than 90% lipoproteins and less than 10% EVs. So it's very important to remove them. So the kit includes two solutions and columns. And the way it works is that the reagents are mixed with plasma, and there's an incubation step for 30 minutes. Then the supernatant is collected and loaded on a prepared column. 1 minute at 1000 G, normal operation for the Vesicec column, and the purified EVs are collected. And what's very important is that the quality that is achieved is really is very good. So the depletion that we observe for lipoprotein is up to 90%, 98%. Sorry. We were still collecting some data, and an article is in submission at the moment, but we have already some data for super resolution microscopy, and the functionality of EVs that have been obtained is very good. As you can see, the size distribution is within the range of EVs. Again, this is a non-dilutive method, and it's because of the easy workflow and the potential scale using this centrifuge. This is a method that we think is going to allow large-scale studies. Another use for Vesicec columns, and it's probably the first one that emerged when the company launched the product, is to clean up samples before flow cytometry or nano flow cytometry, and mostly removing unbound dye and antibody before analysis. So as we know, labeling EVs is not done on a stoichiometric basis. It's very often that we use an excess reagent just to make sure that we have maximum coverage. But that unbound material is creating a background that makes the analysis very difficult, or for some techniques, frankly impossible. The other drawback of unbound antibody or dye is that if it's not properly removed, then any functional assay will see that unbound material staining the cells in competition with labeled EVs. So the experiments won't be conclusive. and the bottom line is that it's quite difficult to remove by ultracentrifugation because it's long and destructive, and gravity size exclusion chromatography tends to be a very long and sample consuming process for such a step. And as we see, this is a very early study that was performed at Aston University, where they have a nanoflow cytometry, where they do they were doing the 2 steps to remove the to remove an unbound dye using the Eisen columns, and that was requiring 2 concentration steps using Amicon filters. And when they switched to Vesisec microcolumns, they were able to drastically reduce this workflow to achieve an equivalent result than you do using the Eisen QEV. And the result is here in the amount of positivity. Without cleanup, only 54% of events are positive for fluorescence. But when you remove the unbound, then there's a better definition of the baseline in fluorescence, and more events appear positive. So over 66%. And another piece of work has been done on the same system, but at Marburg University by Christian Prosser, and he did both experiments using first anti-CD63 antibodies, and without any unbound removal, there was only 13% positivity, and with removal could achieve 22%. And with before removing unbound dye, the positivity was about 30%, and by removing it, could achieve 75%. So that's why the technique has been widely adopted, and some of the publication I will show later to take advantage of this. And that picture shows how the dye retention on the gel, so it's clearly that it's captured at the top of the gel mass, and the product obviously adjuts at the bottom. So as I hinted, there were quite a few publication that came out. As you know, the publication cycle is such that it takes quite a while to come to fruition, but they started to come in earnest at the end of last year, actually. So from September, then there was a steady stream of publication that are including our products, and this one specifically Vesisec Micro. What's important to notice is that they're really a small part of the workflow, but it's more and more visible in terms of the crucial aspect of what they're doing. So, for example, in that study at the University of Nebraska on HEC cells, they make easy use of nanoflow cytometry that I shown earlier, and the sample are prepared using Vesisec Micro before after the labeling with antibodies. And that work was from Imperial College, the group of Paul Freeman and Richard Kelwick, where they use nanoflow cytometry to remove unbound antibody before screening their engineered EVs. And they do the same as well for outer membrane vesicle that they produce as well from engineered bacteria. The study on the top was from the University of Nottingham, where they are labeling extracellular vesicles from cell culture with a dye, with a Vimglo, if I remember well, and they were using ultracentrifugation to do so, which was very ineffective. And by doing it with Vesisec Micro, they achieve a very nice increase in sensitivity. And that on the right, that's another illustration of work that is been performed in Japan, where again there's a contribution of Vesisec Micro before doing analysis of labeled EVs on by nanoflow cytometry. So after isolation, it's important to try to preserve EVs for whatever length of time we need them, and even during analysis. They're very hard to obtain, very precious. So the last thing we want is to lose them because they've been left in the fridge in PBS. As I'm sure you all know by now that keeping them in buffer is leading to degradation if it's left at 4 degrees, and it's a mixture of degradation, but it's also adhesion to plastics. It's been proven that once they are out of their normal condition, probably a change in the protein corona, then they are attracted to plastic surfaces, and this is where we observe a lot of loss during dilution or during storage, during freeze thaw as well. So it's important to stabilize them post isolation to make sure that they don't degrade prematurely. So together with researchers at the Karolinska Institute, we have devised a product called Vesicef, which is really a preservation buffer. It ships in 10 times formula, so it's easy to add to existing samples or just to create from new. And this formula is assisting cryo storage as well as bench work, preventing adhesion to plastics. So the collaborator, the Karolinska Institute, have held EVs for many years, regularly more than 5 years, and the samples have the same properties when they do come out of the cryo freezing. They've carried out extensive studies to show the stability of these EVs that are suspended into the buffer using imaging flow cytometry, and so it's really just to emphasize that this is not lost in time, and the same by doing in dilution. So I'd like to move on to staining extracellular vesicles. It's of very high importance. That's because many of you would want to use techniques like flow cytometry to detect extracellular vesicles, and as we see, it is a challenge to do so, because the the instruments are not always able to function as detection of size of scatter and fluorescence at the same time. So it puts the emphasis on very good labeling that is ascertaining the phenotype as well as the physical properties of the nanoparticles. And I will also emphasize at a later stage the need for reference material to produce an evidence of success when it comes to measuring nano-sized objects. So, oh, excuse me, just another. So when it comes to the labeling of extracellular vesicles, a way to stain all of them indiscriminately is to use a lipophilic probe or dye, and many of them I'm sure you have come to use. They have that's very simple structure where you have a fluorescent moiety. To which are attached long aliphatic chains, and these chains are anchoring into the lipidic membrane. So as you can see here, you have the molecule and the two aliphatic chains anchored into the lipid bilayer of an extracellular vesicle or liposome. You got many commercial products. The most famous is the PKH, but many others use exactly the same the same type of approach to connect to the membrane. The problem with this approach is that it's the anchor is not permanent, and reversibility has been proven in many publications. This means that when that anchor is going, there's less fluorescence intensity on the particle, on the EVs, and that dye is transferring potentially to cells if we are doing a functional assay. It's important to come up with a stain that is more permanent, and this is what we've developed. It's a dye that's called VesiDye LNB. It's from the technology developed by Aquarella in Singapore, and it's intercalating into the lipid bilayer to provide a permanent stain, the one that is very, very stable. So this way there's no loss of intensity and no transfer to cell. And as you can see, the aliphatic moiety is basically in the middle with the length that is commensurate with the size of the bilayer. So there's a whole as you can see, you have different color to cater to very different requirements. It could be for ultraviolet or detection in the blue, in the green. So the FITC equivalent, LNB 520, which is very popular. A long stroke emission 600 that's excited by the blue laser and detected with the red, and we have far red detection as well with the 800. So there's a wide variety of combination of choice, but they all operate in the same with the same principle and the same protocol. And operationally, what's very good is that they're water soluble, so it's very easy to disperse. They don't form micelles or aggregate, which I will explain a bit later, is the issue with conventional lipid dyes. And they are fluorogenic, so they fluoresce only when they are intercalated, making it much easier for to operate. And obviously they have been used in analytics, but also in uptake assay, in vivo and in vitro. So you can carry out your cell uptake studies with flow cytometry, but we have users as well for in vivo in mouse models. What's important to I didn't want to repeat previous slides, but it's very important that even for this dye, it's if you want to carry out any functional study to remove unbound using Vesicle columns, because otherwise despite not fluorescing, the dye will be activated when it's interacting with cells. So it's important to remove the unbound. And as you can see here, it is very efficient when it comes to the low occurrence of fluorescence on the control. You have your your buffer, and when Vesi dye is added, here the LNB 600, then there's very little fluorescent staining from this from the dye, so no aggregate or no micelles. If you use a dye like DI8, which is an anchor lipophilic dye, then there's a heavy formation of aggregates and micelles. It's just because they are very hard to disperse. They are hydrophobic, so they tend to stick together, and they're very difficult to disperse, even if using DMSO, which is not good for EVs or cells, obviously. And when incubated with EVs, so you have the EVs only to the bottom left, and the bottom in the middle, the dye, the LNB dye is providing a very strong signal, very clean signal compared to the DI8. And in the literature, they have been applied again. The product hasn't been on the market for very long, but there's already a few articles out there. This one from that's the Royal Veterinary College in London, where it has been used to on a conventional flow cytometer. I think it was a fax area to stain sheep milk EVs. And on the right, that was from the main group in Singapore, when it was used to that work actually was done by at Beth Israel Deaconess Medical Center, using the dye developed at Singapore, and it was done on EV sample from plasma. So many more applications, but the number of users is quite important now in Europe and the US. So there's quite a few articles in the making. And finally, I'd like to share what can be done in the calibration, normalization of EV analysis using reference materials. As with any techniques, we end up isolating handling material, and we put them through analysis, and we generate all sorts of data. And there's how do we make sure that we have something that we can rely on, and how do we assess multi-parameter capability and achieve orthogonal analysis using so many different variables. So having one material that can be used across a vast array of methods gives a point of reference that is giving strength to this analysis. And for this, we have again teamed up with the Karolinska Institute, where they developed a fluorescent reference material, EV material, and we call it Vesiculab CD63 MNG. So it's MDM green fluorescent, which is equivalent to green fluorescent protein. They are suspended into Vesiculab buffer, so it's very easy to handle. It's just suspended, and as you will see in many of these examples, it makes it very easy to get in nano flow cytometry or EV flow cytometry using any type of instrument sensitive enough. So it's a modern equipment. So on the left, you see that it has been used on the image stream to very easily gate for the population. Here at the bottom right, on the more dedicated platforms, the nano flow cytometer from Beckman Coulter, the Cytoflex Nano. So it's even possible to distinguish the small from the large EVs within the population. So it's very powerful. It's also possible to have a two-color reference by staining with for CD63. So you can see on the top right, that's this is the trace for Vesiculab CD63 MNG, the fluorescence, 73% fluorescent positive. And then when the antibody is incubated with the material, then we have double positivity for CD63 and APC, and that's 70% dual positive. It's also possible to use it for the normalization of physical methods like TEM and nanoparticle tracking analysis, like here on the NanoSite Pro, where you have the signal for the scatter in blue, and then the fluorescence, the fluorescence, which is just underneath in orange. And again, the same experiment that was carried out on the Cytoflex Nano here has been performed on the imaging flow cytometry, the cell stream. And internally, we're using the video drop from Myriad, which is based on interferometric light microscopy. It's a form of NTA that detects the interferometry patterns and track them, and from that signal you can get the size distribution as well as the concentration, and we obtain very rapidly the size distribution. So it's label free, but we don't get the fluorescence from that. And it's possible as well to use SPIRIS, which used to be called the ExoView platform, which is able to capture the events. So the EVs are captured on the chip and further labeled, and it's possible to detect the events that are positive for the CD63 as well as for fluorescence from the neon green. I think we have another. We had someone joining, sorry about that. And in the literature, this reference material has been used in well, first it has been shown in development together with its companion product Vesibind by the group of Samir El Andalusi, where they basically design these EVs for therapeutic applications, and more recently a biodistribution in EV biodistribution study by André Gorgens and Samir El Andalusi has been published to and showing a very nice example of how the MNG EVs are powerful enough to be detected in these studies. And briefly to finish the companion EV from the group in Karolinska involves the presence of FC group at the surface, just concomitant with the CD63 tetraspanins. And this allows the binding of antibody through the FC end, making the antigens available. So this engineering allows these EVs to be used as a template for antibody conjugates, and in the context of flow cytometry to have a true color standards. And as you can see here with the Vesibind and an antibody which is FC, it's possible to have a true color detection. And that's very quickly we also have a lyophilized EV reference material for people who like the GFP, which is a bit more straightforward, but it's a bit dimmer than MNG, and it's there's less positivity for CD63 in the engineering. So that's pretty much it from me. Again, our mission is to make the work faster and more efficient, make EV research more rewarding, and hopefully we can contribute to your success. Thank you very much, and let me know if you have any questions.